Hello guys, today in this video lecture I'm going to talk about the development of central nervous system. So the development of CNS. So let's suppose here is uterus. You can see here, this is uterus. And it has a fallopian tube here. You can see very clearly it has fallopian tube, and this is the fallopian follicle. So what really happens that when the fallopian follicle is fully mature, so the alert surge occurs. A lot of luteinized hormone is released, and then it leads to the rupture of fallopian follicle. Then the fallopian follicle will rupture and the ovum is released. Then the ovum is taken up by the fallopian tube, and it will start moving towards the uterus. Now, the ovum, it has a special covering known as the zona pellucida, and it has the genetic material 23 pairs of chromosomes. 23 pairs of chromosomes. So now the ovum is at the metaphase of second meiotic division. It has completed its first meiotic division, but it hasn't completed its second meiotic division. So if ovum does not receive sperm, then the ovum will degenerate and it will die. So if the scenario is different, or somehow the sperm is come, so the sperm will fuse with the ovum. Whenever the sperm touches the ovum, it immediately becomes active and completed its second meiotic division. So now if I zoom this part, so we can see now very clearly this is zygote. After the formation of zygote, now it has plan to divide. It does not have any intention to die. Now the zygote will further divide into two cells. So now zygote has entered into two cell stage. Now these two cells will further divide into 4 cells. Now the 2 cell stage along with the 4 cell stage is known as blastula. Now after that, the 4 cell stage will further proliferate, and it will make a collection of round about 12 to 16 cells. So this is known as medulla. You can see here. Now this is medulla. Now in the next stage, the zona pellucida is still there, and some cells will make a mass in the center of the cells, and some cells which are present at the periphery, these will start proliferating. So now a group of cells which are present in the center, these are called the inner cell mass. And cells which are present at the outside or at the periphery, so these are known as the outer cell mass. This is inner cell mass, and at the periphery this is outer cell mass. Baby will grow out of inner cell mass, and outer cell mass will convert for the supporting tissue for the baby. Outer cell mass will make covering for the baby, and it will make a placenta. So this is very early differentiation stage, which is inner cell mass and outer cell mass. So inner cell mass is inner cell mass will eventually convert into different layers, which will make the embryo and fetus. And outer cell mass, eventually it will be converting into supporting covering, like we can say like placental tissues. Placental tissues. So in the next stage, what really happens, zona pellucida start dissolving. When zona pellucida start dissolving, a lot of fluid is accumulating with a lot of fluid accumulating there, the outer cell mass remains the same, but the inner cell mass attached at one pole or at one side. So now this is known as the embryoblast. And this one, the outer cell mass, is known as trophoblast. Now after that, what really happens, some cells, so at this pole, at the side where the inner cell mass is present, some cells will start growing up, and it will make a villi. So in the center of the villi, some cells which has intake their membrane, and the other cells which have dissolved their cytoplasm. So the cells which have intake their membrane, these are known as the cytotrophoblast. and the cell which has dissolved their cytoplasm, these are known as syncytial trophoblast. So you can see here, it makes a structure like this. Now after that, the implantation will occur, and it will implant in the formation of the blastocyst, and this is the syncytial trophoblast, and these are the villi cytotrophoblast, and you can see here, this is the inner cell mass. Now what really happens, the inner cell mass is get converted into a bilaminar disc. Now the bilaminar disc has two layers, the epiblast and hypoblast. Now some cells, it goes like this and make a cavity, so this is known as the amniotic cavity. And some cells goes downward like this and make a cavity, this is known as primitive yolk sac. That after that, what really happens, these two layers, these two discs are interconnected with each other at this point, so this is known as the notochordal plate. And some cells degenerate here at this area, some cells degenerates and make a streak-like structure, so this is known as the primitive streak, and it also makes a node here, which is known as the primitive node. If I take out this diagram and draw it here, you can see here, this is the prochordal plate, and this is the primitive streak and primitive node. Now the cell will start moving towards the primitive streak, and it will start moving downward. So it will start moving downward under the surface of epiblast, and it will make a tube-like structure between the epiblast and hypoblast, and it will also make a third layer, which is known as the mesoderm. Now this is, you can see here, this is mesoderm, and below mesoderm there is another layer which is known as the endoderm. You can see here now. So now it has three layers, and it also make a tube which is known as notochord. Now if you see this diagram from front side here, it has ectoderm, mesoderm and endoderm, and this is notochord. Now what really happens, notochord, these cells secrete some growthing factors, and these cells of ectoderm will start proliferating, and it will make a plate-like structure like this. As I said, some cells will arrange like this and will make a plate-like structure. Now this is known as neural plate. Now after that, what really happens, the neural plate, some cells goes downward in the center, and it will make a groove-like structure which is known as the neural groove. You can see very clearly that some cells will go downward, invaginate downward, and it will make a groove, and this is known as the neural groove. After formation of neural groove, some cells will go outwards, and it will make a fold-like structure, and these are known as neural folds. And beside these cells, some cells will completely separate from the neural groove or neural tube, and these cells are known as neural crest cells. Neural crest cells. So after formation of neural groove, that what really happens, these two ports are connected with each other and make a tube-like structure. You can see here in this diagram, this is a neural tube, and these are the neural crest cells which are displaced from neural tube. So these are neural crest cells. So now in the next video lecture, we will talk about the formation of the central nervous system from this neural tube.